are increasingly being able to measure things at a single cell level. Okay, so what's happening increasingly, and today I'll give you an example of the way we measure single cell information in our own lab. What's increasingly happening is that these kinds of matrices are getting replaced with, for example, if I take that this, let me so this is increasingly giving rise to matrices that look like gene 1, gene 2, all the way to gene D. And here, instead of samples, you have cell 1, cell 2, and so on. Okay, so this is really a huge revolution that is currently happening in biology, where we are able to go from the traditional average measurements to single cell level measurements. And this is extremely important because often what happens, and this is the understanding we've developed over the last 20 years, often disease states, for example, arise from very small populations of say 10, 20 cells in these huge populations of billions of cells. Okay, only a few cells go awry, and these are the cells that often lead to disease conditions. So it's becoming possible only in the last 5, 10 years to now start measuring things at the single cell. So this is the current revolution that is happening, and in my opinion, this is going to be the future of biology for at least the next 20 to 30 years. Measurements at the single cell level. All kinds of measurements, gene expression measurements, the kind of things that we've been talking about. Measurements of proteins, measurement of DNA mutations at the single cell level, measurement of what is called epigenetics. There is a whole layer of regulation in biology that happens above the DNA, okay, which is called epigenetics. All these measurements are now becoming possible at the single cell level. These are multiplexed measurements, meaning for any given single cell, you can make multiple measurements of, say, gene expression, protein, epigenetics, DNA, etc. Okay? So that really is the next frontier in biology. And today's talk is going to be about the challenges that this kind of experimental data poses to us. Okay? So it turns out that when you go to this level of single cell, inherently there is a lot of noise. Okay, so if you look at the measurements over here, what you see is that the variation is a lot larger than the kinds of variation that we traditionally see in these kinds of traditional experiments. Okay, and for that, we need a very different approach in physics to start thinking about what can potentially generate data that has so much fluctuations and noise. Okay? And that is an entire field of physics that rests on the ideas of probability, statistics, and stochastic processes. Okay? So what I'm going to do today, the first half of this talk, is give you a glimpse of how we think about stochastic processes that can generate data of this nature, okay, where you have information at the level of single cells. Okay, so I'm going to start and give you a flavor of this thing called a master equation, which allows you to write down the evolution equation of probabilities. Okay, so I'm going to start with a simple example that most of you have seen, and then we'll see what happens when you have such kind of fluctuations that become relevant in these kinds of length scales. Okay, so let's start with a very simple example, which you may be very familiar with. But G, so the example I'm going to take is say gene expression DK. Okay, so what I mean by that is, say you have a bunch of these mRNA molecules that we've been talking about through the course of our talks. So say you have a bunch of mRNA molecules, which I call A. Okay, so A is these mRNA molecules, and with some rate constant K1, they just degrade. Okay, so this is a chemical reaction where bunch of molecules of mRNA with some rate constant ATP. Okay? Where have you seen something like this? I think many of you have seen an example that looks exactly like this in physics. What's that? First order kinetics. Very good. And an example of that? Nuclear decay. Radioactive decay. Very good. Exactly. So if you were to write down an equation to describe its dynamics, what would you write? A first order reaction kinetics. What would you write? So something like, let's say, PADT is equal to what? Minus K1, right? Excellent. Okay. This all of you seem to have seen. What is the solution to this? A as a function of time is just A0 to the power minus K1. Yeah? So what does this look like? If you plot this A as a function of t, this will look like an exponential decay, right? At A0. Very good. So this all of you are familiar. Now if you go and do these experiments in the lab, where you're able to count single mRNA molecules, okay? So here the context is where you can count single mRNA molecules. What you will see is that instead of a picture like this, let's say you do the experiment once. You start with A0 mRNA molecules, and then you wait for a while to see these mRNA molecules degrade. Okay, so let's say you start with 20 molecules. So let's say A0 equal to 20. Okay, what you will see are curves like this. So let's say you have 20 molecules, then one degrades. Okay, so notice what I'm doing. You waited for this long, and then one mRNA molecule degraded, so the number went down by one. Then you waited for some time, then it degraded. You waited for a very short time, then one molecule degraded, and so on. Okay, so let's say this is the outcome of one experiment. You repeat this experiment, what you might see is something that looks quite different. It might look like this. Okay? So what you're seeing, what I'm trying to suggest is that these experiments are not repeatable. Every single time you do this experiment, these curves will look quite different. But what you do get is if you average this, you will get this white line. Okay? So this is at the heart of what we call stochastic biology or stochastic processes, where you can no longer talk about a deterministic phenomenon, right? Because what you're seeing here is that if you look at this time here, let's say this time t equal to t1, you can't talk about a single number, unlike what you could do in the deterministic case. In the deterministic case, no matter how many times you sort of repeat the experiment, at some time t0, you always have some a, okay, which is given by this equation. There are no fluctuations, there is no variation. But here look, at time t1, in one experiment you got this many number of molecules. In another experiment, you got this many number of molecules. Okay? So there is no determinism in these kinds of setups. So the natural language to talk about the evolution of that system is the language of probability. Okay? So the what you can ask is at time t equals to t1, what is the probability of observing n molecules of A? Okay? So this is a very different way of thinking from what you might be accustomed to. Right? So all these kinds of dynamical systems that you saw in the first lecture all dealt with differential equations that look like this, where you are actually describing the average behavior of systems. for these kinds of fluctuations, which become very important when you are dealing with single molecules or, for example, single cells. Okay, here these fluctuations become very, very important. And so the idea is, how do you come up with a theoretical framework that deals with this? Okay, so the aim is to write down dynamics, time evolution equations, time evolution equations for this quantity here, Pn of t. Okay? How does the probability of observing n molecules change as a function of that? Okay, and this is famously known as the master equation. Okay? It turns out that unfortunately this master equation is rarely taught in physics courses, either in the undergraduate courses or in fact even in master's level courses. Only in some special lectures will you come across this. But if you are interested in biology, especially in modern biology, where we are constantly dealing with matrices of this nature, approaches based on master equation become very, very important. Okay, so ideas of probability, statistics, master equation, these are currently some of the most important ideas that are relevant in biology. Okay? So what I'll do is I'll take a very simple example which is slightly more involved than this model of gene expression decay, and I'll show you in a very sort of intuitive way how to write down a master equation and how do you get out of it ideas of fluctuations and noise. Okay, and then what I do is I go to my own lab's research and show you how do you actually measure these things in real cells and what kind of things can you say, for example, about the circadian clock. Okay. All right. So what I'm going to do is a simple model of gene expression. So in this simple model, what we are going to say is that the species of mRNA can degrade just like the way we were describing here via a first order reaction. Phi just represents the degraded species. And the slight increase in complexity here is that what we are going to say is because we have some DNA that's floating around in the cell, and that DNA creates your mRNA molecules, we'll say that in a zeroth order reaction, from nothing, essentially nothing means you have some DNA floating around. And the DNA's concentration is not changing. The amount of DNA in your cells is fixed. So that guy's concentration doesn't matter. So this is a zeroth order reaction in which from the DNA you create some molecules of mRNA. Okay, so this is a very simple model of gene expression. It turns out to be correct only in a certain cases, but it's very instructive to write down the master equation for this to see what kind of noise do we expect from systems like this. Okay, so what we want to do, remember, this is our major aim for the first half. Okay, instead of a dynamical equation that looks like this, which is deterministic, we are going to write down an equation for the problem. Okay, so let's just start writing this down for this example. So what I want is the probability of say n molecules at time t plus delta t. So let me write down one term, and then I will ask you to write down the other terms. Okay, because it's really very intuitive. So at time t, if you had n plus 1 molecules, okay, so what we are going to imagine throughout is that this delta t is very small. Okay, we can arbitrarily choose delta T to be very small so that only one reaction happens in that delta T time. Okay. So that is the situation. What I'm trying to write down, this left hand side, is what is the probability that at time T plus delta T, n molecule. So I'm on the right hand side, I'm going to write down all the ways in which you can get n molecules at T plus delta T. Okay. So the first way in which it can happen is that at time T, you have n plus 1 molecules. Okay. And you have a decay. Remember, the decay happens with rate K1 and it's a first order process. And I should have mentioned the meaning of K1, I should write this down, is the problem. Okay, so let me just mention this. K1 delta T is the probability that one molecule decays between T and T plus delta T. Okay, so that's what these rates mean. So notice once again that we are moving away from these kinds of deterministic pictures. This rate here is that this rate, which has dimensions of 1 by time, so that rate multiplied by delta T gives me the probability that one molecule among the entire species will degrade in this time T and T plus delta. Similarly, K2 gives us the probability that K2 delta T is the probability that one molecule is created. Okay? So now here, this is my first expression. So what's going on here? At time T, I have n plus 1 molecules of this mRNA. This is the probability that one of them will degrade, right? You have n plus 1 molecules at that time. So you have this term here, that's the full probability that one molecule will degrade. Yeah? So beginning from n plus 1 molecules, you have one molecule that degrades, so that gives you n molecules. Yeah, is that okay? This bit is okay. Remember that K1 is the probability of one molecule. Yeah, that's why you have K1 into n plus 1 molecule. Okay. What are, tell me one way in which from time T to delta T you can get n molecules. I give you one equation where you started with n plus 1 molecules. How else can you get here? Okay, very good. So let's say we write this down. At time T, we had n minus 1 molecules. From here, how do you get to n molecules? That? Excellent. Yeah. So K2. So you have a production event. So this was my degradation event, and this is my production event. Okay, great. So you have a production event here. So what would I write here? K2 into. So I'm hearing someone saying K2 into n minus 1. Is that correct? Remember, this is a zeroth order reaction. Right? So it will just be K2 multiplied by delta. So remember, when you have a first order reaction, right, it depends on how many molecules you have in the system. Because K1 is the probability of a single event happening, of a single molecule degrading. So if you have n plus 1 molecules, then the probability becomes n plus 1 multiplied by K1. This is a zeroth order reaction, which does not depend on how much stuff you already have, okay? Because we are assuming that the DNA is constant in the whole cell, right? So this is a zeroth order reaction. So there is a constant probability, right? So even if you have 0 molecules at that point, you still have that non-zero probability of an mRNA being created, right? As opposed to here, remember, if there are no mRNA molecules present, then there's nothing to degrade. Right? So if n is 0, this term becomes 0. Is that okay? So this is the second term, which is the production term. So this bit is my degradation term. This bit is the production term. There is one more term. Can someone tell me what that is? So here you had started with n plus 1, degraded, got n. Here you started with n minus 1, produced 1, and went to n. What's the last possibility? I heard the right answer somewhere that there was n. Okay, so that's a good point. But remember, I told you that delta T is so small, we can arbitrarily take delta T so small that two reactions are not allowed. Only one reaction. Excellent. Yeah, so nothing happens. Right. So the last term is going to be just you started with n mRNA molecules and nothing happened. Okay. So how do I write the term for nothing happened? Very good. 1 minus, what is it? N. Very good. And then there should be something else also.
1 minus K2
Very good. Excellent. So you guys are now masters of the master equation. This is all there is to it. So just to explain what this is what's happening here. If you have n molecules, this term is the probability of one molecule degrading. This is the probability that one molecule gets created. But we want 1 minus that, which tells you that none of these events happen. Okay, so you started with n, you stayed at n. Okay, so that's it. Right, so now if you just take, so notice there is a Pn of t over here. Take this term, take it to the left. What do you have? You have Pn t plus delta t minus Pn of t. Okay. Now notice that there is a delta t in each of these terms. There's a delta t here. There's a delta t here. One has gone to the left hand side, and there's a delta t here and here. Okay. So what I'm going to do is I'm going to divide by delta t. Okay. And so that gives me the following equation: n plus 1, p1, pn plus 1 t plus p2, pn minus 1 t minus n p1 plus p2 pn. Okay, and in the limit delta t goes to 0, what is this quantity here? Pn by dt, right? So you have Pn dt is this whole thing over here. N plus 1, P1 plus 1 plus P2, P n minus 1, P minus NP1 plus P2, Pn. Okay, and that for this particular system is called the master equation. The master equation. Okay. So the master equation is not one equation. It depends on the system that you are studying, but it describes the time evolution of the probability of observing n molecules or n cells, whatever. Okay. So this is a very, very, very important equation that we, in our current research, we are constantly dealing with. Okay. So what I show you in another maybe 5-10 minutes is what you get out of it. Okay. So this was just the simplest, unfortunately, this was the simplest part of it, right? Writing down the time evolution. What becomes very hard is how do you solve this? Yeah. So it turns out that in very few cases can you solve this completely analytically. I'm not going to go into the details, But quite often what you can do is use this equation to solve for what are called the moments. So have you heard of this term called moments of a distribution? So you have, I think most people have. So the moments are simply nothing, simply the average is the first moment. Okay, the variance is the second moment. Okay? So remember, the big goal of moving to this kind of description is to be able to measure fluctuations. Right? What I mean by fluctuations is this, that any given time around the mean, what's the kind of variance that you will see if you measure multiple times? If you run 5 experiments, what's the variance that you will see? That's a large goal of modern biology, right? Where you can measure single cell stuff. So with this kind of framework, you can easily answer questions like that. And I'll just show you how. Okay, so let's see what can we do. Okay, so what we're going to do now, so before that, if you just looked at these equations and you wanted to write down the deterministic version of this, can someone tell me what the equations would look like? So just remind yourself of the radioactive decay problem where we had only this bit. Yeah, but now you have this bit also. So what would this deterministic version look like? So minus, what is it? K1, K. That's it. It will be only this. Okay, fine. Just your constant DNA. Very good. So this is my deterministic equation. So what I first show is that from this formulation, you can get that back. Okay? And then I'll show you how you can get variances out of these kinds of equations. Okay? So the first thing that we do is the equation for the mean. Remember that the equation for the mean should look like that equation up there, okay? Because the mean is what that describes. So how do we do this? So the mean, which I will call M, can be written down as the following. I'm not sure you are familiar with this. So has anyone seen this way of writing down the mean? Yeah, I see a lot of people nodding their head. Okay, good. So essentially all you are doing, you just think about it, is you are averaging a bunch of numbers, but with weights that correspond to the probability of those numbers coming. Okay, so that's the definition of the mean. So then, if you want to write down an evolution equation for the mean, you just differentiate it. Okay, so what you have here on the right hand side is n equal to 0 to infinity. You have n, and then dpn dt. Okay. So what's the next obvious thing to do now? Exactly, very good. You now just substitute. We've written down the master equation here. You substitute it here. So then the next step is you substitute the master equation into this equation here. Yeah. So what will we get? I'm not going to work through the full math because that will take time. I'll leave it as an exercise for you. But I want to show you one trick that you have to use to solve these kinds of equations. Okay. So if you substitute that in, what you're going to get, I was working through this on my slide to here. So you should check that I'm doing this right. So what you get is dm dt will look like the following. I'm going to expand this out because I want to show you one trick that we have to use, which is very important. So all I'm doing is I'm substituting the master equation and I'm breaking out the summation terms. Okay. So what you get will look like this. If I've got things right, Pn plus 1 P plus summation n equals to 0 to infinity n P1 Pn plus 1 T plus a bunch of terms. This is, you can do this yourself, so I don't want to waste time here. Bunch of terms, and then you'll get terms that look like n is equal to 0 to infinity, n a2 p of n t. Okay? So this is what you will get. Just check. This is very simple. But notice now, the problem is you have terms which look like this, p of n plus 1, and you have terms that look like this, p of n. So you can't immediately add up these things. Okay? So what you do is, has anyone seen these kinds of things? No. Okay. So what you do is, you write it down in a slightly different way. Can someone say what we can do? Okay. So what you want to do is basically write each of these terms as p of n. So then you'll be able to add. So see if you agree with me. I'm going to write down only the first term for lack of time. But see if you agree with me. So this becomes A1 summation n is equal to 2 to infinity n minus 1 whole square En. So take a look at what I've done. Where? Now, so what I've done is there are some terms in between. The terms we're talking about, the p of n minus 1, I've not written it here. So, of course, you are absolutely right. So there are some terms that are p of n plus 1. There are some terms that are p n minus 1. And then finally, you will have a couple of terms that are p n. Our goal is to add everything. So we have to convert everything to p of n. Okay, that's one way of doing it. So notice what I've done here. I've rewritten this in a way that this infinite sum still remains exactly the same. Your first term here was 0. Yeah, n equal to 0. So the first term is 0. Then the next term is n equal to 1. This becomes 1. P of 1, P of 2 over here. So I've made sure that that first term is correct over here. So is that okay with everyone? No, because you have, look at the first term that will come here. It will be for n equal to 1, but here you get P2. Right? N equal to 1 will be your first term. First non-zero term. No? Yeah. Just what? Okay. So then it will be, yeah, now in terms of n. Now does it make sense? I think now it makes sense. When n is 0, yeah, therefore you can push it up. Yes, absolutely. Yeah. Okay. Yeah.
You don't have n equal to 0. You can just write n equal to 1.
You can just write n equal to 1. Absolutely. Yeah. So all I'm saying is you have to play these small tricks here for every term and get everything into this kind of form. Okay. So then what you'll be able to do, you'll be able to take out the rest of the terms common. So try this, okay? I've seen that some of you are doing these homeworks. I'm very happy with that. So take this as another homework and show that what you will ultimately get is the following. You'll see a lot of nice cancellations happening, and ultimately what you'll get is something like this. Infinity En of t minus T1 summation N T. Okay? So you get this. Just verify this. Now, what is this sum here? Yeah, very good. This is 1 because probabilities have to be normalized. N can only go from 0 to infinity. N can't be negative because these are mRNA molecules. So this term is 1. What is this term here? M. Excellent. So that's A1M. Yeah. So you've got back your time evolution equation for the average. Okay. But this is very nice because you started with a system which is describing probabilities and you managed to get to the evolution equation for the average. You can do the same thing now. So of course, then you would say, what new have we learned? Of course, at this point, we've not learned anything new. But the power of the master equation is you can do the same thing now for the higher moments, like the variance. So I'll just quickly write down the result, and you can check this once again. So the variance is defined as average n squared minus n average squared. Okay, so that's the definition of the variance. See, this is the second moment coming in. So you do the same thing once again. For example, if you want to write down dv dt, you have to do this quantity here, right? So you have to do d dt of n square average, which is n square Pn of t, and then this thing, right? N average is just this quantity here. That's just m squared. Yeah? And so this becomes summation n square dpn dt minus m squared. Did I get this right? Sorry, there's a ddt here, right? And then you play the same game with this. You substitute in the master equation here. You do exactly the same kind of thing that we did there. And once again, if you're interested, do this homework and show that the equation for dvdt will look like this. Minus 2k1v plus k1m plus k2. Okay. So this is the time evolution equation for the mean. This is the time evolution equation for the variance. Okay. So what this is telling you is what kind of fluctuations do you expect and how these fluctuations would evolve with time. Okay. It turns out that you can also actually compute the entire distribution from these kinds of methods, but we are not going to go into this. I will show you only one last thing, which is very interesting, which is at stationarity, which means when time goes to infinity, okay? So of course you can prove that when time goes to infinity, these distributions will reach a steady state distribution. But at time t equal to infinity, let me call m at time t equal to infinity as m stationary state, and v at time t equal to infinity as the v stationary state. Okay, so these are just the mean and the variance that we solve for at time t equal to infinity. Okay, so what would they look like? All you have to do is set these equations to 0, right? So set equations 1 and 2 to 0. And what you see is that m stationary state is k2 divided by k1. Yeah, that's very obvious. You set this to 0, then the solution for m is just k2 divided by k1, right? And vs, it turns out, if you set this equation to 0, will also be k2 divided by k1. This is remarkable. What you're seeing here is that the mean is equal to the variance. Does anyone know which distribution has this property? Excellent. So it's not just the Poisson, but it turns out that the Poisson is a very important distribution that has this property. Okay. You can also show this directly from these equations, but I'm not going to go into this. But you can show, and of course you can see this here, that the mean is equal to variance, that this gives rise to a Poisson distribution. Okay, so this is actually very nice and has been measured in things like yeast, in bacteria. It turns out that gene expression can in fact be described with a simple model like this, and you actually get mean is equal to the variance. Okay. No, it just reaches a steady state. You have some amount of production of the genes, you have some amount of degradation, so they come to some kind of balance, and you reach a steady state in the mean and in the fluctuation. To observe it for a longer time. Exactly. It's a stable fixed point. So if you perturb it, it will come back to the same steady state. Yeah.
So it's the energy of the flow rate, that's the production would always be higher than the degradation.
Not necessarily. It completely depends on the biological system of your interest, right? So you have some production, you have some degradation, and you can see that the balance of these two will allow you to reach some non-zero steady state. So you will get these non-zero steady states, and you can measure these fluctuations, and you see that this is okay. It turns out we can do this, and this is where I'll connect to my research a little bit. You can do these measurements, and I'll show you the kind of measurements you can do for the circadian clock genes. And the very interesting thing is, circadian clock genes turn out not to be Poisson. Circadian genes are not Poisson. Okay. So this is very interesting because this comes out of a model where in some experiments you do see that Poisson property holds. But if you actually measure the circadian clock genes, and I'll show you the data in just a bit, it turns out that they have a property that they are super Poissonian. Which just means that the variance is larger than the mean. Okay? And it turns out that the distributions that describe circadian clock genes is no longer Poisson, but what is called a negative binomial. Okay? The distribution is called a negative binomial. So it turns out that most genes, in fact, are negative binomial distribution. And what we've understood from this kind of a theoretical approach is that the way this can happen is a slight modification of that simple model that I showed you. Remember, this model of A going to 5 and 5 going to A, this gives you Poisson. So how do you get a negative binomial, which is what we are interested in, which is the circadian clock case? It turns out that for a negative binomial, there is something called a telegraph model, which is a simple extension of this idea, but to a concept of two-state gene expression. So the idea then is that this process is happening, but in the presence of a switch, meaning you have a protein that comes binds to the DNA, and only then you start getting these molecules. Okay, and every once in a while, this molecule falls off, and when it falls off, there is no production of A. Okay, so this process where, so remember, this bit was only described here. But now you can add this information that a protein molecule is required to be bound for this kind of mRNA to be produced. And it turns out that you can show that this model, in fact, indeed gives you negative binomials. This is exactly what is telegraph. Okay? So now what I will do, since you've got a sense of this, maybe 10 minutes, I'll move to my laptop. I'll show you some of these kinds of measurements, and I'll show you what the implications of this kind of negative binomial sort of super Poissonian noise is in our measurements of circadian time. Okay, so that's where I want to finish. Yeah. You can set out the laptop.
So when you talk about decay from 8 to 5,
Yeah.
DNA is being converted to mRNA, right?
No, then I'm talking about the mRNA decaying to some nucleotides.
Is this kind of spontaneous decay?
Yes. So it's not really spontaneous decay. So what happens is for mRNA, there are particular proteins that come and chew up these mRNA. So I'm not describing all that in so much detail, but I'm just writing this down as A goes to 5. But of course, there are proteins that come and chew up these mRNA. Okay. Very good. So no, so it turns out that this fact that the circadian clock genes tend to be distributed as a negative binomial. This is an intrinsic property of that network that creates the circadian clock. So it doesn't matter who you're taking these. Yeah, absolutely. Right. So you'll see that right now, the data we collected, even from the same different cells, you'll see that the parameters of the negative binomial.
Let him. What is it?
So I guess in the meantime, if there are any questions, you can ask me. If there are any questions right now.
So
Yeah.
I have a question. So by solving this master equation, we are going to feed the value in the covariance the PCA for finding out the parameters and stuff? Broader sense?
So no, not really. So you can do that, but what we usually do is that covariance we construct it directly from the data set. But you're absolutely right. If you want to do pure theory, then yes, you can write down the master equation. You can then write these kinds of things down for multiple genes, construct the theoretical covariance matrix. That's one way of doing things. But what I'm going to show you today is how we directly measure these things in the lab. What's that? Sorry. Oh yeah, so exactly. So these are different techniques. So the standard way of doing, so for example, if you want to figure out the parameters of the negative binomial, so the negative binomial has a few parameters. You have these switching rates. So let's say you have K1, K2, this is K3, and so on. What will happen is the negative binomial distribution will be parameterized by these three parameters and also this one will be defined. Yeah. So your distribution ultimately the negative will be parameterized by these parameters. Then we can measure the distribution in the X that I showed you. Then by putting these distributions to the real experiment, then extract. This is all not okay. This is all bad. First of all, yeah. So these three expressions, this negative one and telegraph is the most. I don't think we have recently go beyond that. Already there are too many parameters. So experiments determining these parameters itself becomes a challenging problem. So then if you go even higher non-Markovian, maybe it doesn't make so much sense. Yeah, so I'm okay. So the way we do the experiments, maybe because this is taking time, I'll just explain on the board itself while this is coming up. Should I also maybe email it to one of you? Yeah, can I do that? Yeah. Screen sharing. I'll share it on WhatsApp. Okay. So while this is going on, what I'll do is I'll tell you how these experiments work. Okay, I'll give you the conceptual basis of the experiments so we can directly see the data. So the way my lab works on these experiments to answer Professor Seva's question is a method that's called single molecule fish. Oops. Q. fish. And what fish stands for, it's a long title, it's called fluorescence in situ hybridization. Okay? So it's actually a very simple idea, but very powerful. Because what you can do is imagine you have a bunch of cells in your dish. Okay? So these are all single cells. This is the nucleus. Okay? So what we want to measure is the mRNA molecule. Okay? So let's say you have a bunch of mRNA molecules of yellow color floating around. And you have a bunch of, say, pink molecules floating around. And your goal, remember, is to measure the number of pink mRNA molecules and the number of yellow mRNA molecules. Okay? So what we can do is we can design primers, what are called primers, which are essentially, perfect. How many of you have heard of the term Watson-Crick base pairing? Okay, some people, okay, not bad. So what happens is, if you look at DNA, you have base pairing, A pairs with T. So DNA is made of what are called bases, A, T, G, and C. And double-stranded DNA has this kind of pair. Okay, A pairs with T, G pairs with C, and so on. So you get these double strands. So the way these experiments work is that synthetically we outsource this to companies. So companies will come and for this yellow molecule, which looks like this, say the sequence is A, G, C, C, A. That's it. Okay? Usually these are about 1,000 base pairs. So it's 1,000 base pairs, but the sequence looks something like this. What we do is we design primers that have the complementary base pair. Okay, so A will complement with what's called U, G with C, C with G, G, U. Okay, so this bit we actually buy from companies. So they synthetically design this and give it to us. But in addition, what they do is they stick a fluorophore at the end of this molecule. Okay? So now what we do is we flow these guys onto the cells. We permeabilize the cells so they become permeable. Then we flow these guys into the cells. So these primers go and bind wherever there are these yellow mRNA molecules. And remember that they have the fluorophore bound to it. Okay, so each of these guys have fluorophores bound. Now, if you take it to a epifluorescence microscope, so there's a technique called fluorescence microscopy. So you shine a particular wavelength of light on this, it emits a different wavelength of light. You can collect these signals, and then what you will see is that in these cells, for example, in this cell, wherever there was a yellow mRNA, you see a spot of this color. Okay? So here you had two mRNA molecules, you see two spots, one spot there, one spot there. Okay? And these are diffraction limited spots, and this you will probably have learned in your optics course. I remember all this, airy discs and all of that. So these will all be diffraction limited spots, airy discs, with a width that is set by the frequency of this one's emission. Okay? And you can then use machine learning algorithms to go over thousands of cells like this, compute the number of spots in each of these cells, and then you can look at the distribution of the number of spots over all these cells. Okay, and what you will get are these distributions. Okay, any questions on the experimental technique? So this process takes about 2 days. You have to take this and hybridize this method of complementary base pairing. This is the hybridization part of fish experiment. That takes overnight. And then what you have to do is imaging, right? You have to take it to the microscope, and these are require extremely sophisticated microscopes because you're trying to detect single spots, right? So you need very fancy microscopes that are cooled and so on. These are LED microscopes. So that also takes a little bit of time, but the major time is that overnight step. So roughly in 2 days, you can get experiments out from this. And so the first thing I'll say is I want to thank the students who did these experiments. So most of these experiments were done by these two girls and Anju, and then Shabnam did all the machine learning, etc., to count these spots. So this is the technique that I was talking about. And I don't know if you can see this. So this is the kind of data that we get. So these are all single cells. Maybe this one is the clearest. These are single cells, and you can see these spots here, right? So this should really blow your mind. Each single spot here is a single mRNA molecule. Now, if you think about the kind of length scales involved, one mRNA molecule is of the order of 100 nanometers in size. Yeah. So you are being able to detect single molecules of that small length scale with these kinds of microscopy techniques. Okay. So you can see, and what I've shown you here is you can multiplex. Again, remember I was telling you that the frontier in biology right now is to be able to do multiplexed single cell measurements. So what you can see here is 6 different genes that we have measured in the same single cells. Okay, and what you will get out of it are these distributions, what we just described, negative binomial distributions. Okay, so it turns out that if you look at the statistics, these are not Poisson, as that simple model had predicted, but these are all negative binomials. These are the fits, and from these fits you can get out the parameters, right? So anyway, so I'm going to try to end very quickly in the next 5 minutes, but the problem with this now is that because these circadian clock genes are so variable, meaning the variance, remember, is greater than the mean. That's the definition of super Poissonian noise. So then the challenge becomes, at the single cell level, is there even any meaning of circadian time? Remember, the whole lecture series was on measuring circadian time, right? So we went into all this mathematics and ideas of fluctuations because we want to ask the question, is there a concept, a notion of circadian time at the single cell level? Okay. But the problem is we have so much noise that comes simply because of these fluctuations in the chemical reaction. So with this kind of noise, is it even sensible to talk about a circadian time? This was a big open question in the field of circadian clocks, and we've been working on this question. And what we think, and this is results that we are hoping to publish very soon in the maybe next few months. I will skip these details, show you just this slide. What we do is from these distributions, we have a few machine learning algorithms that we've developed, through which we can try to infer what the phase or the time is, right? Just the way we learned in yesterday's class. Now we've developed new techniques to do this for single cells. And what you're seeing here is basically the number of cells required to give these kinds of errors. Okay. So what you're seeing, I'll walk you through this slide. These are absolutely new results. Okay. So which is why this lecture is about the future. If you take a single cell and many single cells and try to predict the circadian phase or the time, you see that the errors are all over the place. And this is happening because the noise in gene expression is super Poissonian, as I've been explaining all this while. Okay? Notice that errors are all over the place from 0 to 12 hours. That's the maximum amount of error that you can make. So it's uniformly distributed. But what we are showing is that even if you start averaging over a few cells, these errors in your phase inference can drastically go down. Remember, when we talked about phase inference and time inference yesterday, we were using millions and millions of cells and 10,000 genes, 500 genes of that order. Here, what we are showing is after you can do these very sensitive experiments at the single cell, the single cell information does not give you any time. Okay, it's all over the place. But even if you average only 20 cells, you can see how the error has gone down below 1 hour. Okay, so this is quite remarkable. What this is telling you is that you don't need millions of cells. You can just use 20 to 50 cells to get these kinds of very accurate measurements of circadian time. Okay, and this is a comparison. Here, this is data once again generated in our lab. We are using just 3 genes, okay, as opposed to those kinds of thousands of genes that have previously been used. 3 genes, once again, you see the average errors we are getting is of the order of 1 hour. But here, this is the state of the art currently. You can see that for this kind of error, you need over 10, 15 genes, and these are all using millions of cells. Okay? So we are being able to push the boundary to make these measurements using only 3 genes and with as few as 20 to 50 cells. Okay? And the additional advantage of these things is that we are able to do this with spatial resolution. Okay? So I just want you to take home this message that biology in the next 10, 15 years is also becoming very spatial, meaning you have not only information of these single cells, but you also want information of these single cells in the context of space. Because, for example, if you take a tumor biopsy from your liver, it's a solid piece of material, right? The spatial location of your cells can make a very big difference, whether it's close to a artery, whether it's close to another biological structure. These things make a huge difference. But the methods that I talked about yesterday are completely agnostic to space, because what you do is you grind up all the cells and you lose all geographical and spatial information. But these kinds of techniques can be done in intact tissues. Okay, so you also have spatial information, and what we showed in another experiment is that you can get these kinds of spatial information out, spatial circadian clock time out of these kinds of experiments as well. Okay, so I've clearly run out of time by a lot, so I'm going to stop here. But one final thing that I've talked a lot about to all of you is that if you are interested, if you got interested in these kinds of ideas of using physics in biology and you want to pursue it, for the first time, we have a program in India jointly between the International Center for Theoretical Sciences, which some of you may have heard of, ICTS, and NCBS. It's a TIFR program where we are inviting physics students, mathematics students, and computer science students to come and work on these really hard biological problems. Okay, these are some of the most exciting problems now in the field, in science in general. And if you want, you can come and work on these kinds of interdisciplinary problems which require modeling, theoretical ideas, machine learning, experiments. And so you can visit the website and see much more of this in detail. These are currently we have about 12 faculty on board. And the fun bit, which I want to end with, is that 4 out of this 12 are ex-Stephanians, okay? All from the physics faculty. So Sandeep Krishna, me, Archishman, and Akshit. All of us are from this, we studied in the same room here, okay? So you can also be one of us if you want. Thank you very much.
Thank you so much.
Thank you.
I'm sure all of us learned something new and really good. Now
like to request our yes, just gave us deliver.
Thank you.
Popli Memorial lecture series. And as we already said, it's a flagship event. And we really wonder and plan who the speaker should be over the and this is because we have a lot of things in mind whether the speaker can motivate, encourage, inspire the students because this is one of the platforms to do that, or to give a flavor of different kind of research or new research that's going on, or to know about fields that they may have tread upon or not tread upon, or to apply their concepts and knowledge to understand what is being developed, and also to take in what is new and excite them into these fields. So, keeping all this in mind, sometimes we are not really able to achieve this, but this time, thanks to you, that we are able to achieve most of this and almost all of it. And thank you, Shaon, for not only introducing us to circadian clocks that each one of us have, but also entraining us to your clock. So that everybody will, alert throughout the whole thing. So each one of us has been taught by your lectures, and I'm sure after this, people would like to explore and discover what their own clock is and try to explain this accordingly. So thank you for giving your time and enlightening us with your research. You said it was surreal to come to college and become the speaker. It is also a moment of pride and joy for all of us to find you back as the youngest speaker of the Popli lecture series. Thank you so much for your kind support. And at this point of time, I would also like to thank Dr. Chakraborty, who was kind enough to help us conduct a dance workshop. She is an eminent dancer, of exponent of Bharatanatyam, and in fact, she has just now given a very, very good news. She has been awarded the Ustad Bismillah Khan Yuva Puraskar for 23 by the secretary. The workshop was very, very successful. She gave her creative inputs, ideas, and her dance steps, nuance steps, and insights to the finer details that could help the society, the students who are interested in dance, encourage them. And so, thank you, both of you. And it is like, nowadays the marketing gimmicks are like you want ek ke saath ek free. Have the Popli speaker also additional credit advantage. Thank you both of you for at this point. I know the entire evening so much very feel the tent feeling, but there's an army of people who actually work through it, put it, and I would like to call students up. I know there is entire team, but I would just call upon the heads who are dealing with it. So, Nadia. At this point that they were also guided by the advisor, Dr. Hari. Society as family of physics department. We are really grateful for the help or encouragement from the principal. Thank you.
Request Sangeeta ma'am to present a moment to Dr. Chakraborty. I request her to hand a moment to Dr. Jayaraman. Now, as it is the third and final day of the Popli Memorial Lecture Series, the Society Journal Echo is officially released today. It is my pleasure to invite the journalist Alice Samir to present the journal to Dr. Chakraborty.
We would like to present our annual journal for Physics Society of this year. The first 20.
Oh, thank you. This is going to be regular.
Yes. So that brings us to the end of this year's 11th Popli Lecture Series. Thanks to everyone for your kind presence and attention during the lectures and for making it a success. I would like to request all of you to gather outside for